Record. All right. Well, thank you everybody for being here. Just a reminder that we're recording this session. It'll be posted and available later. If you would like to see the transcription or captions during this session, you can go to the menu bar, mine's on the bottom, and click on More and turn on your captions or the transcript on the side if that provides greater accessibility for you. Otherwise, thank you so much. I'll turn it over to Sarah to kick us off.
Great. Thank you so much. And thank you all for being here and giving us the opportunity to talk about an unconventional topic for a conservation biology meeting. This is a great venue for this project. So before I get too far along, oops, I went too far. I just want to give a little bit of an introduction to myself and then give my co-presenter a chance to introduce himself as well. So again, I'm Sarah Orlovsky. I'm at the University of Wisconsin-Stevens Point, and I'm a parasite and disease ecologist, and I also teach aquatic ecology. I teach a lot of our undergraduate kind of field methods courses. And just a little bit of background, I came into parasitology through the lens of conservation. I was very passionate about amphibian conservation about the time that BD, the amphibian chytrid fungus, was coming to light, and amphibian malformation. So I started my career very much with parasites are the bad guy, and we need to figure out how to protect amphibians. And now I've kind of turned the page and hopefully will inspire some of you to kind of take a broader perspective of parasites at the ecosystem level and kind of see them as part of an integrated approach to conservation. So that's a little bit about me. I'll turn it over to Robert and he can share a little bit of his background as well.
Yeah, my name is Robert Jaden. I work at Lawrence University. I'm an evolutionary biologist, and in particular, I work on systematics and phylogenetics. And so I'm involved in describing new species and figuring out evolutionary relationships and sort of thinking about biodiversity from a broad perspective. But part of that also incorporates sort of the thinking about these lineages and their persistence and not going extinct. And so conservation is an important part, I think, of evolutionary biology. And then also, as we're thinking about conservation of species, we have to think about what is a species. And so one of the things that I do is describing species as delimiting which different populations might be differing species and which ones might actually need protection. And so I'm sort of interested in that perspective from a big biodiversity point of view, but also narrowing that down to various groups. Most of my research is done on snakes, but I also work with my collaboration with Sarah on parasites as well.
Thank you, Robert. And we're going to kind of go back and forth. So we'll kind of take a slide here or there, but feel free to kind of interject and ask questions along the way. We don't want this to be a lecture. We want this to be very interactive. Our goal is for you to come away with how to use this info and the assignments and assessments in your classroom. So please feel free to stop either one of us along the way if you've got questions. So Robert, I'll have you take over the overview here, and then we'll kind of go back and forth.
Am I able to? Okay, I think you have to. Yeah. So the workshop overview that we're going to talk about, we're going to give a basic background on parasites and conservation, why they're an important group to study in this aspect, how they're the utility that they could have in a classroom, along with the sort of motivation for this particular project and the learning objectives that we have for this classroom activity. And we'll talk about some of the components, and sort of Sarah mentioned in the promo for this is sort of this can be diversified in a lot of different activities. And so you could take this one sort of project idea of assessing particular parasites as are they in need of conservation or not, and you can make that into oral presentations or written reports on in a variety of different outreach activities. And so that's one of the ones that we'll be talking about as well, how you can direct these different ways. In Sarah's parasitology class in particular, she's actually put this into practice, and she's gotten a lot of great feedback from students after she's actually completed this. And so we'll see some of the sort of student outcomes, but also how the students receive this particular these assignments, because it's a really neat sort of concept, and in a lot of ways I'll talk about a little bit in a bit, but it kind of goes against sort of what our initial thought process is on what parasites are and how they interact in our environment, how we should perceive them. And a really important part of education, as we all know, in that when students sort of struggle with something, they learn a lot more. And so parasites in particular kind of get this different mindset where it's like you got this really you're doing the switcheroo on something, and when you have that, I think the students learn a lot. They have this idea of this is the way the world works, and parasites kind of throws that on their head already. But then in addition to that, now let's make parasites the good guy, where they can actually be things like bioindicators, and they need protection, and that's going to provide a lot of sort of critical thinking and mindset change, and that's really important in education. Which there's not a whole lot of topics I think better in parasitology to do that for students. And then so we'll talk about questions and discussions so that you might have and some input that you might have as well to make this or to make this maybe better, or any ways you could think about adding your own spin to this. I look forward to hearing about what you guys come up with. And then also, like Sarah just mentioned, happy to slow down and reiterate things, or to have a fresh perspective. If you guys have any questions along the way, just let us know.
All right, so I'll jump back in, and I just want to show this beautiful compilation of parasites. So when I say the word parasite, certain things jump into my mind. When you hear the word parasite, something very different might jump into your mind, and that's because parasitism and is a very broad life history strategy. So it's not a taxonomic group. There's not one evolutionary lineage that consists of parasites. There's multiple sort of convergent evolutionary events that have resulted in parasites. So I wanted to start with a little bit, hopefully not too much, of Parasitology 101, just to help folks, remove some of those barriers, some of the jargon, some of the terminology that we have. So these are all parasites. So Robert's going to come back with the definition here.
Yes, so like Sarah was mentioning, parasites, when we think about parasites, a lot of our students might not think about this question or where they belong on an evolutionary tree, but from a phylogenetics perspective, it's kind of a life history strategy. So basically, a parasite is any organism that lives in or on another living organism, that's sort of the host, and obtains from it part or all of its nutrition or needs of existence, and imposing a net detriment to that host. And so we sort of think about that from an elementary perspective, but when students really get inundated with this sort of broad concept of what a parasite is, and first from an evolutionary perspective, that if you look across the animal kingdom, the parasites have evolved numerous times, even within various groups. And so it's not just sort of, students often don't just think about, they just sort of, oh, a tick is a parasite, and a particular worm might be a parasite, but not realizing that life history strategy of basically obtaining nutrients or housing or something like that from the host is very beneficial to a species. And there's a lot of evolutionary reasons why you might become a parasite, and it's happened so many different times across the evolutionary tree of life. And so that in itself is this really cool concept that really gets students thinking about big scale evolutionary macroecological patterns. And so Sarah's mentioning and bringing this up in a parasitology course, but you could teach this one little section in general biology, and it sort of puts a big framework as to an important lesson in science in general. And so that I think this could be adapted to a lot of different courses. So even just a basic biology course could really improve, or could use this lesson. Yeah.
All right. So in terms of diversity and abundance, and that first slide kind of showed this, and I like to use this quote in class as well, is all living species are involved in parasitism, either as parasites or hosts. So we have yet to find a parasite-free host. As much as humans try to become parasite-free, we are plagued by about 200 of our own species that live in or on us. And because of that diversity and abundance, each animal is typically host to more than one thing, that parasites actually outnumber hosts, even though we spend a much smaller proportion of time thinking about them. So the estimates differ, and this is in large part because we don't have a handle on parasite diversity very well. Parasite diversity is still under-described, but based on our current knowledge, about 50% of all living plants and animals are parasitic at least at some stage of their life cycle. So it's a pretty broad topic. If there's a favorite host or ecosystem that's your passion, there's probably a parasite that can be put into it. So this is something that I think is really critical to the activity that we're going to talk about today. And you'll notice in that very wordy definition that we use, we tried to avoid the word harm. And many classic definitions of parasitism use the word harm, but I'm going to try and explain why that's a really kind of slippery slope. So parasite does not equal disease. And that's the first kind of misconception to get through to students, is that having a parasite doesn't necessarily make you sick. Many hosts are very infected and yet don't show overt signs of disease. And so why is that? And how can we kind of wrap our heads around that? Harm is also very difficult to quantify. We're really good at understanding what's healthy, but it's very difficult to describe disease in all of its different facets. This is species and even individual specific. I get a cold and I get the sniffles for a couple days, and other people will be in bed for a week. Your immune system is different, your genetic constitution is different, your environment, nutrition, all of those things determine whether or not you get sick. And last but not least, and I work on worms, and so some of you might be familiar with this. This image came out in the early 2000s when we were investigating flatworms as the causative agent for amphibian limb malformations. But you can see in the top image, this is a frog that's infected with a parasite. It doesn't have very many, but the one on the bottom does. So it's the same species of frog, same species of parasite. But what's important is that for many parasites, we have intensity dependent pathology. That means the more of them you have, the more likely they are to cause pathology. One parasite, you're fine. 10 parasites, maybe you're not feeling so good. 100 parasites, you are in the danger zone. So this is a really important concept, and I'll expand on this because it's really not relevant for things like viruses or bacteria or even fungal diseases, but for some of your larger parasites like your worms and your arthropods, this is the rule of the game. So this is a really busy slide, but I wanted to put this here. This is from the textbook I use in parasitology. But this addresses some of the differing perspectives. If you've taken a microbiology course, or maybe your students have taken a microbiology course, they have a very different definition of parasites than a parasitologist would, or that an ecologist would when we talk about parasites. So this kind of puts them all together in terms of their taxonomic groups and some of the terminology, what we call a microparasite versus a macroparasite. What's a pathogen versus a parasite? So I'm not going to go into too much detail other than to acknowledge that in the different disciplines, there are different perspectives. So if you want some hands-on advice, I know some of the other speakers said that they would be happy to meet outside of this session and outside of this event. I'm more than happy to kind of work with folks to try and see where this could fit into their course or help their students based on what background they have to kind of conceptualize this and understand, well, when we're talking about parasites, who are we exactly talking about? So I'm going to dive into the micro versus macro parasite distinction because that's going to make the activity go a lot better. Microparasites, these are things like viruses and bacteria and protists, and they multiply within a host. These cause acute disease. These have short generation times and are the diseases where hosts tend to develop immunity. So they kind of function more like your typical common cold or maybe the stomach flu. So those are kind of what we're thinking of for microparasites. For our macroparasites, these do not divide and reproduce inside a host, and they're accumulated individually through these very distinct transmission events. So they function very differently biologically in their hosts. And these are the ones that, as I mentioned earlier, have intensity dependent pathology, where the number you have determines how sick you are. So I talked about this in the promo, and I'm going to expand on it here because I want to address that idea that folks come in with, that parasites can extirpate their hosts and parasites pose a threat to conservation. That is absolutely true. I don't want to dismiss that. I don't want to sound like parasites aren't harmful when they are. We know that this is, if you're familiar, this is an example from the Hawaiian Islands with the birds that have faced enormous pressure from avian malaria and the introduction of avian malaria with mosquitoes to the Hawaiian Islands. But I do want to point out that when we think about threats to conservation, they're typically those microparasites. They're viruses, bacteria, and protists, fungal diseases. So there's that distinction. And when we think about changing perspectives and parasites being beneficial in the environment, potentially being indicators of environmental health and providing benefits to ecosystems, we're typically talking again about macroparasites. Things like worms and arthropods tend to be in this camp. Again, this is not black and white, but those tend to be the distinctions. Oops, I went too far. So this is not my original idea. I'm not the first one to talk about conservation of parasites. I'm just interested in this topic coming from a background in conservation biology. So this was a review paper that's come out a couple of years ago. So scientists are beginning to recognize that parasites warrant conservation along with free living species. So this has been a call to arms in the parasitology community and also extending into some of the conservation biology literature. So right now, this is in the very conceptual stages of designing kind of what it would take to do conservation for parasites. And that's where I'm going to turn it back over to Robert, who's going to talk about some of the ways that's been happening.
Yeah, so one of the big pushes that's been going on for a while is to sort of categorize and to classify all these different species that we know about once we've described them into these different categories of conservation concern. And so the IUCN Red List is one of the main organizations that's doing this to sort of assess all the different potential species on the planet and see how are they doing and over a long time period and sort of getting snapshots of their sort of environmental health, population health over time. And so you've probably noticed, you know about endangered species, but you probably also know that there's different degrees and levels of endangered species. And so, but your students might not. And so if you look at this sort of diagram at the bottom left here, you can have different categories of species. We know that some species go extinct and we know that some are endangered, but there's a lot more categories than this. We've got things like least concern. We're not worried about this species going extinct anytime soon. There's no real threats that we've sort of located for this particular taxa, like white-tailed deer. You know, we do population viability analysis on that species, and there's not much of a chance of it going extinct anytime soon. Whereas other species, there's different levels of population concerns, and with different sort of impacts that the environment has on them, or whether it's human hunting or habitat destruction or disease, something might be declining these populations. And so there's everything from near threatened to vulnerable to endangered. And then when we get into endangered beyond that, we get things like critically endangered, extinct in the wild, where we might have these in captivity, in zoos. We had a lot of this with the frogs in Central America and some South American species as well, where chytrid fungus went through and basically made a bunch of species extinct in the wild. People had collected them with the Houston Zoo and Zoo Atlanta for the amphibian ark, and they brought them into captivity. And so we still have those species, but in the wild, people haven't found them after sort of chytrid fungus went through. And so one of the things that the IUCN does is sets up all these little groups, these specialist groups of these sort of main experts in the world that assess all the biodiversity. And so in my case, I mentioned I study snakes, and so I'm on the pit viper IUCN species specialist group, and we look in a number of us, maybe a dozen of us, go through and say how many species are there of these species, what's their distribution, Are some of these species cryptic diversity species where there might be more than one species hiding in what we consider one species? What's the range? Is that habitat getting destroyed? Is there a pet trade issue where people are illegally taking these things? And so we've got assessments for that for mammals and birds and all kinds of other groups. And so we've had over 93,000 animals assessed for IUCN Red List. Some of the species that I've described are on this, and yet it's very vertebrate biased. And as you'll notice here, there's only two species that are parasites, not actually worms, just a louse and a flea that have been assessed and sort of allocated to being on this list. And one of the benefits for a species to be on this list is it can hopefully restrict if a species is on this IUCN Red List, it can be part of the formation of the information that they have for like CITES, so the international trade of endangered species, and so they can ensure that people aren't able to distribute various individuals that they find. It can restrict research on particular species. It can restrict habitat destruction sometimes. And so the IUCN plays a major role in that. But first, you need to have those species assessed. And so the big issue also is data deficiency. I didn't mention that, but that's one of the big challenges with species in general. We don't have a whole lot of biologists studying all this biodiversity, but parasites in particular, not only do we not have the many biologists studying them, but it's also really challenging to study those guys as well. But of all the species assessed, only two have been parasites. Sarah, do you mind going back one more? Sorry. And so the goal in this is to compile all that data, assess that, and then review and revise those submissions, and then submit that to IUCN as a committee. Go. Yeah. And so, as I mentioned before, there's some taxonomic impediments. How many species are there? Are these legitimate species? Is there more species than we think? And then because some species, especially with parasites, get described based on their host, and there's not a lot of molecular work. And then sometimes the morphology is very challenging. A lot of these species are really old. The old descriptions aren't the same as new descriptions for a lot of taxonomy. And then a lot of species are data deficient. And there's a bunch of challenges with that. And Sarah, you might have some additional things to talk about with this, perhaps. Yeah.
Yeah. So as Robert just described, the biggest problems we have with parasites is being able to describe all the species and being able to have data beyond the descriptions. And IUCN has the guidelines for dependent species. So most of that work has been done on mutualists rather than parasites. So we have to adapt it a little bit for that. But the barrier that I'm going to kind of springboard off of is the barrier of public opinion. And this is where we took what the IUCN is trying to do, and that's really kind of the crux of our activity is before people are going to do anything about parasite conservation, we need to change their minds about parasites. And again, I've acknowledged that there are some parasites that cause health and economic issues for humans, domestic animals, and wildlife. But it's a small minority of the parasite species that we know about. Diseases and parasites are not synonymous, and disease is mediated by these host factors. And many parasites are not, in fact, harmful to their hosts under the most circumstances. And this is where I really kind of had my change of heart as I started studying in graduate school the roles of parasites in food webs. And so you can see here, this is a food web that contains parasites, and understanding how parasites interact with those ecosystems. So we've been talking a lot. We've been kind of moving through this pretty fast, but I'm just going to intentionally put this slide here to remind myself to pause and just ask if folks have questions so far about these issues or the background or something that's confusing before we move on to the activity itself.
I do have one thing to add. Sarah's mentioning a couple times about how parasites do cause some harm. But from an evolutionary perspective, when you look at the ones that do, for example, chytrid fungus that we brought up that had all the big impacts on amphibians in Central America, that's an old world fungus, it seems, and it only became really problematic when it went to a new novel place, and those species hadn't evolved to deal with that. And so same thing with the avian malaria. We're used to malaria in our species being about 4, maybe 5 species of malaria. Um, but the majority of malaria species are in lizards and birds, and most animals do just fine having that malaria. But when they get to the Hawaiian Islands that never had malaria, and those birds have never evolved an immune system to deal with that, then it becomes quite problematic. So even the parasites themselves that we think about as being problematic tend to often be problematic when they are invasive or introduced. And from an evolutionary perspective, those novel hosts are not prepared to handle those parasites.
Thank you, Robert. All right. Not seeing any questions in the chat. I can't see all the hands, but feel free to interrupt us as we go along if anything is unclear. All right. So now we'll get into the teaching part. So I teach in upper level biology elective and animal parasitology. This is a lecture lab course, and I have a really unique opportunity because most of my students are in either wildlife management, conservation biology, or captive wildlife management. They want to work in zoos or aquariums. So my interests complement those of my students. They're very interested in working on conservation topics, either in zoos with captive wildlife or in the field. So they already kind of come primed with the conservation background, but they come also primed with let's get rid of all the parasites. Parasites are bad mindset. So this assignment was really motivated to address that misconception that all parasites cause disease, and also have a fun and creative project that improved students talking to each other and also students talking to the public, because this is an ology. We have practical exams. They spend a lot of time on microscopes. It's daunting. So this project was meant to kind of give the students a lot more creativity, which is something, again, we've talked a lot about at this conference, give them a creative outlet in this class. The other part, and Robert, you can add something here too if you want, is we then take some of what the students create and use this as public engagement. So we like to share about parasites with different stakeholders. We work with a lot of hunting groups and with our museum to educate the public. So maybe there's some aspect of your classroom or public engagement where you can use this activity to kind of foster those interactions as well. Robert, did you want to add something to that?
Yeah, we get quite a few of our specimens from hunters, and so we look at a lot of like Sarah's research on duck parasites in particular. And so people get really excited when they're invested in a particular study. And so when they hand Sarah a duck, or sometimes we've had fishermen as well, and then they sort of see this animal that they're ready to eat, and they're used to just sort of processing this themselves. And then they come back and see Sarah a year later, and Sarah says, "Oh yeah, your duck had this kind of parasites in the intestines." They think that they're not thinking that these things actually have those parasites in it. So it's kind of neat to actually outreach to the public where they see this animal, they just assume that it doesn't have anything, or they don't think about it, and it's kind of a neat. And then they can also, with Sarah in this picture, you can see the microscopes there. So we'll often bring slides or vials and things like that with us as we talk about these parasites to these duck hunters or to the public in the museum, and they can actually observe and see these things that normally people don't get to see. It's really neat. It's sort of a different world that most people don't get to observe. And so again, that's another aspect of changing people's mindsets. And then when some kid's saying it's gross, you can kind of contradict that a little bit and saying how neat it is also, and they kind of get a different perspective. Yeah.
Although I will say it's mainly older folks, it's parents. Kids love the microscopes and and all of that stuff. They love the gross stuff. So maybe it's because they're still at that age where that's exciting to them. So as I mentioned in the promo, we had 3 sort of broad learning objectives for this assignment, and that was to get the students to learn about a real life host parasite issue using primary peer-reviewed literature, evaluate the conservation status. So that's why we provided that background on IUCN. We did not want students doing a full-on assessment. If that's your goal and you're really excited about that, please let Robert and I know. We're undergoing this process, the training for the IUCN specialist working group. We're not on it yet. We just learned about it this summer at another conference, but we can point you in the right direction. So This was motivated and inspired by that IUCN Red List, but it's not meant to be an assessment, although you could do that. We wanted to use that framework, though, to motivate the students and help them make that connection. The goal for us was to have them write something that they were excited about and then share it with their peers through both oral and written communication. So I'm just going to pause again here. I want to make this, not a lecture, but just some thought questions for you to take a minute or two to kind of jot down, what made you come in this workshop today? What brought you here? What is your motivation for this topic and the assignment? What course or courses could this be incorporated into? Many of you are probably teaching something in conservation biology or maybe ecology, so it might take on a different flavor there. What background on parasites might you need to have for your students or for you? What is the foundation you need to start? And then what are your learning objectives on that continuum from sort of outreach, or are you interested in that assessment? So I'll just give folks a minute or two here to kind of think about the answers to these questions, or to jump in with questions for Robert and I before we continue on to the nuts and bolts of the assignment.
I'll just add one thing before we get started again, Sarah. So before you get started, let me know.
I actually set a timer because I know if I don't, like in my classroom, I will just keep going like a train. Just all right. So that's about a minute or two that we'll just pause and kind of let you brainstorm. Also, at the end, we'll have a longer time for Q&A, and we'd love to get your ideas on how you might use this to help us improve it. So, Robert, you said you had something to add before we continue.
Yeah, another benefit of looking at parasites is, when we teach like phylogenetics, the history of evolutionary life in general biology, we'll often try to teach phylogenetics by looking at the evolution of vertebrates, where you go from fish to amphibians to reptiles to mammals and birds. And it kind of looks it's really simplistic and it makes intuitive sense. And so we get a lot of students who think they understand phylogenetics because they've studied that sort of terrestrial vertebrate evolution, and therefore, but then when you actually try to get at the mechanisms, do they really understand phylogenetics? They often don't. Similarly with conservation, when you talk about conserving things like giant pandas or tigers or something like that, there's this sort of thing that people sort of intuitively understand, and the research it takes to get to understand, should this particular big charismatic mammal get conserved. Everyone's on board. It's easy to get the information. But with parasites, it's kind of really neat that you can actually, you really have to dig in deep and understand the different hosts and the ecosystems and the complexity of this problem, which makes it a lot more, I think, again, impactful from a teaching perspective to really get at people's, the real underlying aspect of conservation, and to sort of hit some more neurons than the normal typical conservation of pandas and stuff. Yeah.
Yeah, we had a great workshop yesterday on the value of species. So if you attended that one, that was fantastic. And then and if not, check it out in the recordings. But yeah, he had a suite of 10 different species and asked students to rank which ones were more valuable for conservation. And he actually had a parasite on there too. So for some of the same reasons, to kind of make people more aware of those interactions and kind of the knee-jerk responses that some of us have towards different species or others. So Awesome. So the assignment itself, we're going to dive into the nuts and bolts here. So what I'm going to do is tell you kind of what the assignment includes and then discuss some of the teaching notes, the implementation, kind of the advice I would give behind the scenes. So the first part is the actual assignment, and this requires students to identify a threatened or endangered host. And if you were here at the very beginning, Jessica, our moderator today, and I were talking about co-extinction. And when a threatened or endangered host goes extinct, the parasite goes extinct as well if it's a host-specific parasite. So that tends to be the most fruitful area for students to search and start their search and have the highest likelihood of finding a successful match. So it helps them understand the co-extinction crisis and gives them a good starting point, kind of narrows that search down. They search for scientific peer-reviewed literature. Now, I'm dealing with an upper level biology elective. If you're using this in a lower level course, maybe you can use this as an opportunity to have a librarian come in and talk about, searching for peer-reviewed literature, how to do that, how to do a literature search. My students already know, but it's good to refresh on that. And then they collect data, again, inspired by the IUCN and Red List criteria, but not in a strict quantitative sense. I don't expect students to estimate population sizes for the parasites or effective population sizes for the parasites. That's a little outside of the scope, but you could do that. But it boils down to the life history of the host and the parasite, the distribution, abundance, and other traits that relate to conservation status. And then they write a short, publicly accessible summary. So something around the range of 500 to 1,000 words, something that someone could read in a few minutes, because we all know that our attention is taken in a lot of different directions. So trying to make something that's appealing to the public. So I'm going to go through a lot of text here. This is, you don't have to write this all down. I'm going to share the assignment with our moderators so that they can post this in advance. So I'll just kind of run through what the assignment looks like for the students. So they write about the host biology and the threats. And so this is one way to have them learn more biodiversity. The parasite taxonomy, what is your parasite? The potential host parasite interaction. So in some cases, the parasite is harmful and they find an endangered host where the parasite is part of the threat. So that's not off limits, and that's important to talk about as well. And then they can kind of grapple with, is this parasite found on any other host? Do we know if it's on any other hosts? Is it on other endangered hosts? And so this is where they kind of get into the details of that interaction. The parasite biology. So many of the parasites that I study have complex life cycles. So they are in 2 or 3 or 4 different hosts as part of their life cycle. So is it simply one host that's endangered that can be the cause of endangerment? What is the geographic distribution of each of the hosts that they need to have in their life cycle? So this gets into some more of the details of the parasite part of the story. And then based on that evidence that they've accumulated, this is where they have to make a judgment call. And this is where I know that there's no kind of strict criteria. You could implement a more strict criteria. But for me in this assignment, I just wanted the students to kind of wrap their head around, well, what would you consider? If this is an endangered host, is the parasite endangered and why? Can they make recommendations for how to preserve the parasite along with the host? Is conserving the host going to be adequate for conserving the parasites? As I mentioned, many of my students are interested in working in zoos and aquariums and in wildlife rehab. So they're excited about the conservation potential and maybe not delousing everything immediately when it comes into a rehab center or something. To that effect would go a long way into preserving parasite diversity. So in terms of implementation, how I could see doing this, of course, you guys can have other ideas as well, and we'd love to hear them. But I did this individually. I have a class of 24, so that seemed like a good amount of different profiles for me to read. Maybe you're in a larger course and you want it to be in groups, or you want the group interaction. I do a lot of other group projects in my class, so this was a good opportunity for students to go off individually and pursue something of their own interest. The mechanism for choosing the parasite and host. I made my students approve it first because I didn't want a whole bunch of duplicates. I knew I was going to get dolphins like crazy or sea turtles because those are always favorites. So I wanted a good amount of diversity. So I had it be kind of a first come, first serve. You could have a list of chosen species you want them to work on, or maybe you're focused on a geographic region. You want them all to be in the desert, or you want them to be in a particular habitat. Maybe your local habitat could be good, but some kind of way to help them choose and help you feel like you can assess them kind of on an equal playing field is important. The numbers and types of scientific sources, for many parasites, they're only going to have 1 or 2 papers because that's the species description. So I usually keep the numbers of required sources pretty low, but they will find a lot more on the hosts. So make sure that this fits your learning objective, something to consider in terms of what's out there. And then your specific target audience. Mine was other undergraduate students so that they could present to the peers. But maybe you're getting ready for an outreach event and you want your students to engage with the public and you know something about the particular people you want them to engage with. So you can kind of build this around that. And then I did the written component as well as sort of an informal kind of present in groups to each other. If you wanted them to do a formal PowerPoint presentation, conference style presentation, that could be fine too. It depends on how this fits into your particular class. In terms of having run this before and from the parasitology perspective, these are kind of words of warning or tips for success is limit this to those macroparasites because when they start getting into viruses and fungi, that's when you're going to find more pathogenic organisms and it's much harder to establish the criteria for a population of a virus or a bacterium than it is for an animal like a louse or a tick. Exclude parasites that are known to cause disease, like just about everything has some sort of cryptosporidium. They're everywhere. So I'm trying to put some boundaries on that. Choose species that have been described and one thing students run into is there's a flatworm in this. Well, that could be in a family or an order. You have to be narrow enough where they can find out what they need to do. Terrestrial and freshwater parasites are much better described than marine, and parasites that have more than just a species description will be more fruitful. And again, those hosts being threatened and endangered, that is really the key to helping them kind of learn more about the hosts as well. So part 2, after they do the written part, is each student brings in a printed copy or brings in a laptop and they meet in groups of 3 or 4 and they sit around and they take turns kind of presenting to each other. And this is their highlight of the class. This is kind of the best day of parasitology for many of these students because they love sharing their pet parasite with the class. In terms of implementation, set a time limit because they are so excited, they will literally take the whole time period to do this. Monitor the groups and make sure that everyone has a chance to present. If you mix up groups, make sure everyone gets equal time and plan in advance if there are student absences. If someone, I teach in Wisconsin in the winter, the chances of there being a snow day are pretty high. So do you record things and put them on Zoom? Do you have a virtual option? So just kind of keep in mind if you're doing an in-person presentation, kind of how to navigate that with those small groups. And then last but not least, this was the reflection. So they had a second writing assignment, and this was to, after they did kind of the round robin portion, to talk about the commonalities they had between their parasite and host and others they heard about, and talk about the conservation issues that they encountered and kind of how to fit all these pieces together. Implementation wise, I would recommend having students give feedback to each other. I didn't do this, but it's something you could do if you're most interested in that aspect of students giving kind of peer-reviewed feedback to each other. Or if they are giving this to the public, include that in their reflection as well. So mine was simply for this first iteration just done in class with other students, but these are other ways that you could do that. So I'm going to, again, kind of pause here for a minute and give folks a chance to kind of digest the components of the assignment. Again, we'll post this once we have the means to do that. So you can have the assignment in front of you, but think about things like the size of your class. Would you do this as individuals or in groups? If you are doing it as a group, should you have a group contract or some sort of mechanism of group evaluation so you can hold individuals accountable? I did this over about a month's time where the different parts of the assignment were spread out, but do you want to do this just in a week, or is this a semester-long project? How in depth do you want to go? What type of presentation and peer feedback? What other assignments in your class does this complement, or can it coexist with? Do you write for the public? Are you interested in communication skills? Is this a good avenue for that? So again, this doesn't have to be a silent time. If you've got questions, let us know. But I'm just going to set another minute timer just to kind of give folks a chance to take some notes and have some thoughts, and please share questions if you have them. Awesome. So thank you, Martha, for the question in the chat. The question is, I'm curious how often parasites are host species specific, e.g. lice, because I wonder if delousing is a community health action for zoos because it could cross hosts. That is a fabulous question. Lice are among the most host-specific ectoparasites. So lice, even on a species of bird, you'll have a distinct species of head lice versus body lice versus wing lice, and they don't interact with each other. A lot of that is determined by grooming. So the beak grooming, the size of the louse and the feathers are so specifically attuned that if the louse is too big or too small, the bird can groom them off. So lice are very host specific. The other end of the spectrum are fleas. Fleas will bite anything. So I completely understand like getting rid of fleas. That would be a huge problem in a captive environment. Lice are a little bit different. And so that's a great example of where the students can get into the biology and understanding the actual host-parasite interaction and how that might make a difference on how you treat that species. Also fleas, larval fleas exist in the environment. They're in the bedding, they're in the feces of the host, so they can contaminate bedding material and habitats, nesting material, things like that, versus lice never leave their host body. So if you're thinking about control and treatment in a captive environment, those things come into play. So yeah, that's a fabulous, fabulous question. Awesome. Any other questions before we kind of wrap up with the last kind of segment of our presentation today? All right. So what did the students think? This is why I'm here, because they love this. I wasn't sure what to think the first time I did this, but the students really loved this project. They loved having the creativity of choosing their own species. Maybe it was a favorite species. Maybe it was something they wanted to work with in the future. Maybe it was something that inspired them to love nature. They really loved sharing with each other. This was a great team building activity. In my future course, this coming fall, I'll be doing this earlier in the semester as almost like an icebreaker so that the students can get to know each other a little bit better. But overall, the reactions to this assignment were confronting their own biases about conservation. We've talked a little bit about that value and how we value some species more than others, and also understanding the threats to conservation. New awareness of parasite diversity. I think a lot of students thought there's like 200 parasites and I'm going to learn them all in a semester, and that's just not not kind of the full picture. The recognition of the merits of at least certain parasites for conservation. And again, and what I hope to convey to this audience is that they understood host conservation better. And the risks that hosts had and how that fits into the larger ecosystem. So I wanted to share just a couple of student comments. This was from their reflections, and they gave me permission to share these. I just was kind of blown away by some of the thoughtfulness that the students had. So this student reported that discussing the research project process with other students helped expand their knowledge of parasitology. Parasites are not a topic that are set in stone and studied to completion. Just as I'm learning about parasites in class, people are out there discovering more and learning along with me. Though this is true of most subjects I'm currently studying, the number of resources that have been allocated to them is significantly higher in comparison to parasitology. So just becoming more aware of like the knowledge gap, that taxonomic impediment that we have in parasitology. And this student shared, as a wildlife major, I've always loved animals and try to stay up to date with the many endangered animals in the world, but never considered parasites in this. When this project was explained, I understood how it makes sense that host-specific parasites could go extinct with their host, but I never thought about it before. Additionally, through this project, I learned how understudied parasites are, which is surprising to me. So just kind of, open-ended prompts of, tell me what you learned from talking to your peers about this project, and they came away with some pretty important learning outcomes. So how do you grade this? You might be wondering. So I have rubrics again in the handout that I'll share with the group after this meeting. I have rubrics for both the summary and the reflection. I did not grade the in-class portion. I was kind of milling about the room, kind of being the vulture, circling, listening in on the conversations. So students didn't really need a carrot or a stick to do that. They just loved talking to each other. They were engaged out of interest and wanting to share their story, and there was enough of an incentive to talk to their peers for the reflection piece that part didn't have to have an actual grade attached. In terms of my improvements for the future, my point values were low, so in the assignment, keep that in mind, and I might actually make a note of that when I post it. I wasn't sure how high stakes I wanted this to be in terms of the proportion of like how much this assignment made up in the class, having it be kind of the first iteration. But the students put a ton more work into this than I thought they would in terms of the writing and the peer-reviewed literature searching. So I would just caution to make this assignment commensurate with the amount of work that they put in. Students really do spend a lot of time writing, and peer-reviewed literature searches are hard for obscure species that students like to pick, like rare birds in Australia that I had never heard of. So that's all for the formal presentation today. I put mine and Robert's contact information there if you want to reach out to either or both of us outside of this group. But I'm happy now to kind of open this up to the room and answer any other questions or get your feedback on this assignment and think about how we can do this better in our classrooms as well. Yeah, Stuart.
Hi, thanks so much. It was a great workshop, really fascinating stuff. I wanted to kind of go back to Robert's point he made earlier about that sort of native, non-native species dimension. And just curious if that has played into the discussions or what students are submitting. I teach a course on invasive species, and yeah, that dimension I think is pretty fascinating. So I'd love to hear if that's come up in more detail.
That's a great question. It did not come up in terms of this assignment, but I think that's because of kind of the kind of tight reins that I put on the choices of the parasites and the host. I think there's an opportunity where if that's what you're interested in, this could be very easily modified to be pick a threatened or endangered host and investigate if it has an introduced parasite or not. I think that could be another spin on this. There's a lot of adaptability there. As Robert mentioned, elsewhere in this course, we talk a lot about parasites and introduced species. We actually have a whole chapter of the textbook devoted to that, both how hosts that are introduced move parasites around, and also how parasites that move to different environments can have potentially more negative effects on those native species. So I think while it didn't come up directly in this first iteration, I think that this could be easily modified to either have that be one of the learning outcomes, or it could definitely come up just with student choices in the future. So that's a great connection there.
Yeah, it can be a really good perspective as well to say, like, I study snakes, and Guam is a very famous scenario where, as somebody who cares about the conservation of snakes, I would still eradicate all the snakes on Guam because they're leading to a lot of birds dying. They've made like several species extinct and all that. And so it's a nice sort of contrast with like, here's parasite conservation, here's some examples of some parasites that should be conserved. Let's say you've analyzed that as a classroom and say, but then we've also got this sort of alternative as well, where we actually need to actually, you know, have some impact on getting rid of some parasites as well. And so, biodiversity conservation isn't just sort of one, let's kind of conserve everything, but there's some dynamics that in order to conserve some things, we might have to manage and get rid of some other things. So, for example, the brown tree snake in New Guinea, that's fine, but the brown tree snake in Guam, let's get rid of that as fast as possible. And so parasites are that kind of interesting group as well that can sort of teach that lesson.
The other, I'm just going to jump back because Stuart's question is so fascinating to me, but one of the reasons that zoos are starting to become more aware of, like, maybe we shouldn't get rid of all of the parasites of this species is because when they put them back, hopefully for release, there might be other parasites there that their immune systems would then have to encounter. So we need to kind of prime them for release if that's the conservation goal, because we know that there could be introduced parasites in that ecosystem, things that they haven't encountered before. And if they're naive to that, we're doing the species a disservice by not maintaining at least that evolutionary relationship to parasitism that might help them succeed in that environment.
Yeah, they've had that issue, I think, with the black-footed ferrets, where they basically eradicated all the parasites for black-footed ferrets, and then whenever they try to release them, they just get inundated with parasites, and so it's not a good conservation strategy for the ferret to get rid of all the parasites and things. And so it's a really interesting dynamic of some parasites are important for them by themselves, some we should try to get rid of to save hosts, and some are actually a necessity for conserving particular hosts. And so that real, there's no black and white with any aspect of biodiversity. And so highlighting all that gray area is going to really improve their learning and teaching and stuff.
Yeah, it's such a great topic. It really just highlights all that complexity, and there's so many angles to it. Yeah, it's fantastic.
Thank you. I appreciate that. Yeah, I'll be thinking more about those introduced ideas, the species introduction ideas too, and how that can play out in this assignment. And I want to thank Cassie too for her feedback. Thank you for the comment in the chat. And I agree. Yeah, working with large primates, that's another issue where some folks have a lot of misconceptions and sort of, kind of people either love them or hate them, and there's a lot of sort of misunderstandings about those species as well.
I can speak this in too.
Yeah,
Yeah,
go
sorry,
ahead.
Jessica, you go ahead.
Oh, I was mentioning before we got started, just chatting a little bit with Sarah, that I am a few doors down from a parasitologist who's come and given her pitch in a couple of my classes for why we should care about parasites. And so I found that it even fit into like I teach an environmental ethics course where we talk about the value of species and what species we give moral consideration and what kind of ethics underlie that. And so, even in a very kind of non-sciencey course, it was really interesting to bring in this kind of perspective when we think about species and how we consider them, how our laws protect them. The students were like, this was a topic where they just had never thought about it or recognized the importance. And the co-extinction piece was really like poignant for them to recognize, given that there are so many host-specific parasites. So I didn't introduce the topic myself, but just she came in and presented it, and we had a little discussion, and they really, it was kind of eye-opening for them and made them even question like their own values around species and conservation, where they were like, oh, I didn't realize I was so speciesist in my in my own values. So that was an interesting extension of the conversation.
Absolutely. I couldn't agree more. That's fantastic. Oh, Martha.
I just want to jump off of what Jessica just shared and some connections that I'm making on this. I mean, I shared in the chat that I could see using this the kind of start of a class, but one of the other interests that I have is also in kind of bringing in other voices. So I love that you do that, Jessica. And then I'm thinking, Stuart's going to do one on podcasting. Could we have a parasite podcast? That would be, or are any of your students creating materials that I could use in my class that then have a voice and face that's not mine, that maybe is more relatable because it's more age appropriate, or they represent, they have identities that I do not hold or that are not commonly held, and that they could then go, wow, parasitologists of color unite, would be really cool.
Yeah, that's a great idea. There are some parasite podcasts, but they're very biomedical. They're very case study related. It's most of them are we found this weird parasite in this host. What is it? And they kind of and they're great. Don't get me wrong. I have friends and colleagues that do that, but I agree with this perspective too. Is the biodiversity piece. Just parasites in general from a wider perspective than the medical perspective would be great to get more voices and more perspectives in this area in particular. So, yeah. Great. Well, I could keep talking about this for forever, but I think we are about out of time. So if there's any other questions or comments, please do feel free to reach out to Robert or I, and we'd love to hear from you if you've got feedback on these assignments. And then Martha, I'm not sure who will be reaching out to us, but we'll be happy to share those assignments and so that they can be posted and accessible to folks.
Great, thanks. Yes, Bernie will reach out to you, and everybody will have the link to the folder where they will exist.
Great, thank you.
Thanks so much.
Thank you very much. Thank you.